And I will time you to 10 minutes.
Yeah, so I was gonna try and find you earlier and I could not get that presentation under 10 minutes.
Huh?
I was like, I cannot get under 10 minutes,
So what is it? 12 minutes?
like 15. So,
That is fine. I don't want you to rush, but
okay.
that grade will be
Yeah, that's
dinged
fine.
a little bit.
Okay, good.
Yeah, go ahead. I can put this so you can use that with a pointer. Pointer should be working. Do you want to use here or do you want the, what do you prefer?
I could use, I guess, the room.
Yeah, so yeah. Yeah.
Okay.
So big.
Okay. Hello, everyone. To start off the presentations on the effect that protein fractionation has on structure and function, today I will be presenting on the effect of extraction pH on heat-induced aggregation, gelation, and microstructure of protein isolate from quinoa. So to start off, why is this not working? What is it doing? Oh, okay. Oh, okay, yeah. So to start off, why quinoa? So to start, quinoa is considered an Andean grain that has gained a lot of popularity recently due to it being its health promoting benefits. And since it is considered a pseudo cereal, it is also gluten-free. So a good source to look into. One of those health promoting benefits is it's a good quality protein, ranging from about 12 to 23%, and has a really pretty balanced amino acid profile that's better than most cereals and legumes. Additionally, going specifically looking into the protein, quinoa is composed of 2S-albumins and 11S-globulin, known as chemopodin. And this is of interest because this globular protein is similar to the globulin, the 11S-globulin in soy, glycinin, which has high functionality. And then quinoa protein isolate is commonly extracted using alkaline extraction isoelectric precipitation method, which is widely adopted in industry for the production of protein isolates. So to kind of set the stage, previous findings include of quinoa protein isolates that extracting increasing the extraction pH results in a higher protein content and yield, more protein denaturation, changes in aggregation, dissociation, and protein structure, as well as lower solubility. Additionally, quinoa comes in two different varieties, sweet and bitter, and commonly bitter quinoa has a high level of saponins. And most studies that have been done on quinoa protein isolates have been done on the bitter variety. However, there has been studies done on removing the saponins from the bitter varieties, but removing the saponins showed to have a decrease in functionality. Additionally, previous studies had looked specifically at cold-induced gels and not heat-induced gels and the resulting aggregation and gelation properties of that. So this study aimed to kind of look at heat-induced gels, which brings into my objectives, which is first to look at and determine the impact extraction pH has on QPI properties, kind of comparing it to literature, such as purity, yield, solubility, molecular weight distribution, as well as the heat-induced properties, aggregation, gelation, and the gel microstructure. Additionally, the study also aimed to understand how the functional and physiological properties are impacted by the variety of sweet quinoa being naturally present low in saponins and determine if this factor has an impact on QPI. So to get started, preparing the QPI, they first had a little bit of sample prep, which included milling and sieving the seeds, and then followed by defatting using a 24-hour sock slit method with petroleum ether. And as we have learned in class, defatting is important in order to have an increased efficiency of protein extraction. Next, they followed the alkaline extraction method, which we should all be kind of a little bit familiar with. But to kind of go through that briefly, the defatted flour was suspended in DI water. The pH was adjusted to 8, 9, 10, and 11. This suspension was stirred at room temperature for 4 hours and then overnight for 16. The mixture was centrifuged in order to separate the protein-rich supernatant from the fiber and starch. Then the protein-rich supernatant was precipitated at pH 4.5 to collect the protein, centrifuged to separate the protein out, and then resuspend this protein-rich pellet in water in order to help remove some of the salt. And this is important again because salt can interfere with the structural and functional properties of the protein. Then the protein-rich pellet was separated out again, resuspended in water, and neutralized to then be freeze-dried. So the methods of analysis that they conducted during this experiment was first noting the protein yield and purity, the molecular weight distribution using SDS page, solubility over a range of pHs at 1% protein solution, the particle size over this same range of pHs, the thermal properties using DSC, and then looking at the effect of particle size, but looking at the particle size over a range of temperatures, and then as well the effects that heating at the same range of temperatures has on the gelation properties, and then imaging the microstructure of the gel using CLSM. So the first thing they looked at was the effect of pH on protein yield and purity. And generally what they noticed that by increasing the extraction pH, it resulted in an increase of protein yield. And this is to be expected as we know that solubility at a higher pH allows for more ionization of OH and SH groups, which then can increase the interactions with water. Additionally, they saw a decrease in the amount of protein lost to the grain fraction during the alkalization step, which is inversely proportional to the yield, kind of showing that more protein was lost at a milder pH. And then again, that with increasing the extraction pH led to a decrease of purity. Next, kind of looking at the molecular weight distribution, the main thing to kind of look at is this 11S globulin protein at around 50 kilodalton. And so they performed the SCS page under non-reducing conditions and not under reducing conditions, but had noticed that at pH 8, 9, and 10, that the globulin fraction was more preserved versus at 11, you kind of don't see it here anymore. So they concluded that at pH 11, either the protein, this globular fraction was hydrolyzed into lower molecular weight fractions or had formed insoluble aggregates. Additionally, because of this, they saw more banding present at the higher pHs, and they also thought that the higher pH could be more successful at obtaining lower molecular weight fractions. And the reason why I have this question mark here is because as per their method for running SCS page, they centrifuged the suspensions and collected the supernatants and ran the supernatants on the gel. So if there were insoluble aggregates present at the higher pHs, those probably would have been removed prior to loading it onto the gel. And I think we can also see this at the top here, because at the lower pHs, you have more proteins of larger molecular weights kind of sitting there. But at the higher pHs, you don't really have that, which you would expect if there was more insoluble aggregates forming and would show a sign of like polymerization. So next I looked at the thermal properties and noticed that most of the fractions denatured at an average of around 97 degrees Celsius, and they largely attributed this to the globular proteins, as most globular proteins and other grains denature around the same temperature. However, as you can see here at E11, they did not have an endothermic peak, which kind of goes back to the SDS page, where they didn't have a strong globular fraction present. Additionally, they didn't really notice a difference of enthalpy of denaturation, or sorry, the denaturation temperature with extraction, but they did notice a difference in the enthalpy that kind of seeing here again, because there was no peak, it showed that pH 11 was very denatured versus a milder pH showed less denaturation, having a higher enthalpy. Oops, sorry. Okay, next they looked at the solubility and particle size. And as you see here, this kind of graph at the top should look really familiar because as we know, globular proteins tend to be pretty insoluble around their isoelectric point. But overall, because the pH E8 and E9 tended to kind of preserve the globular fraction, they concluded that these milder pHs also could have helped show a wider range of solubility over a wide pH. However, they also noticed that, as we know, because an increase of denaturation would cause the formation of insoluble aggregates due to hydrophobic interactions and also hydrophobic regions being exposed, the more denatured protein isolates would have had a less decrease in solubility, which was also kind of seen here. Additionally, when marking the particle size, you would expect that their isoelectric point would have the highest particle size as the proteins are aggregating, which you kind of see at least in the pH 8 extraction, but you don't really have that same relationship here. I would assume that it would be like an inverse curve of particle size. And so I would also attribute that back to their method is that they filtered the samples prior to running the particle size so that any insoluble or large aggregates that were forming maybe at these pHs probably were not accounted for. So finally into gelation, first thing that they noticed was that the onset of gelation or the increase in particle size really occurred starting around 60 degrees Celsius, but mainly for the 9 and 8 extracted QPI. And so this was shown like the increase in particle size kind of aligned with maybe the formation of soluble aggregates that we know is very key in forming a gel. However, for the higher pHs, they didn't notice that increase in particle size, meaning that these soluble aggregates are not forming. And again, due to their denaturation state, probably just form aggregates within themselves that could not associate together. And again, going back to the higher enthalpy from the more milder pHs, we could see here that going into the gelation, that obviously was a lot higher, and that kind of can kind of assume that there were more disulfide linkages being formed. And especially here at this time, at this temperature is around 70 degrees, which again, kind of going back to the DSC, the enthalpy of denaturation kind of like started, or the endothermic peak kind of started around 75 degrees, which marks the onset of denaturation. And we know that some denaturation and some protein unfolding is key for gelation, so that way you can have induced protein-protein interaction and protein-water interaction. So they also noticed that as the graph kind of started getting more exponential, is going closer to that denaturation temperature. But however, again, looking at the higher pHs, they didn't notice this increase of viscosity until the temperature ramp was cooling. So that kind of concluded that like these extraction conditions were not able to form a gel during heating, but only upon cooling. And this could be due to the fact that, again, due to their low solubility, and the insoluble aggregates probably sedimented before they can interact with each other. Additionally, all of the gels basically got firmer upon cooling as well. Okay, lastly, they looked at the gel microstructure. And so kind of going back to the gelation results, you see a lot more non-protein sections in the milder pHs, which is probably going to be water because the protein is encasing water or holding on to water. And that is indicative of a denser protein gel matrix versus at the lower pHs, you have a lot smaller pockets and it's not going to be as dense of a protein gel matrix. And you just have a lot smaller particles at these higher pHs. So, in conclusion, they were able to establish that, again, the extraction pH does have an effect on purity and yield, denaturation, molecular weight distribution, and solubility. Additionally, they were able to establish relationships between the extraction pH and heat-induced gelling behaviors, and thought that a milder pH QPI could be applied for semi-solid foods and be used for a gelling application. And that harsher pHs, because they did not gel, could be applied to a beverage and other liquid-based applications. However, I have this question mark here because as we saw, the solubility was not as great for these harsher pHs, and their formation of insoluble aggregates would not pose them up well for a liquid application. So in the future, they also said that we'd have to explore more applications for quinoa protein isolate, as well as refine the extraction method for maximized purity and yield. So for some of my own critiques, some pros of this paper is that they try to approach this with an industry focus, as previous research only studied cold-induced gels versus this one is heat-induced, which is more aligned with how industry would process applying heat, as well as studying a sweet quinoa variety in order to eliminate any sort of impacts saponins might have had in the functionality of quinoa. However, I feel like as an objective of theirs, they kind of lacked on this mark. Because if they were comparing their results to literature results, which I assume were also from bitter quinoa varieties, they accounted these differences in results due to their extraction technique difference. And as we know, the technique of extraction will greatly impact your structure and function. So it would have been better if they had a control of including like a bitter quinoa variety going through the same process and a sweet quinoa variety going through the same process to be able to compare like if the difference is due to the absence or presence of saponins. Additionally, it would have been nice to also have studied surface properties like charge and hydrophobicity, as they were likening the solubility to a decrease in overall net surface charge and an increase of hydrophobic regions being exposed. And just having that data would have kind of confirmed that theory. And lastly, like I mentioned, for particle size and SDS page, they filtered their suspensions prior to running these analyses, which kind of greatly impacted the assumption that there were aggregates present that was causing this underperformance in functionality. So what about the applicability of this work? To me, the extraction process seemed really long and impractical for industry. It was 20 hours long. So again, they would also need to improve the process also for yield and efficiency, because if you're looking at the more milder pH, which performed better for a gel, that one had a yield of about 35 to 40%, which probably wouldn't be great. And that's on benchtop. So it's bound to be reduced, scaled. Additionally, studying solubility over wide range pH kind of was unnecessary, as we all are familiar with that solubility will decrease around the isoelectric point, and most food applications are either at a more neutral pH or an acidic pH, like a high acidic beverage. Additionally, they defatted using petroleum ether, which to my knowledge is not GRAS method of defatting in industry. So it would have been better reflection if they had used hexane, because this could also have an impact on the protein denaturation, as defatting can expose hydrophobic regions and impact the structure. But finally, they were able to establish that their gels at a 10% protein solution were stronger than pea and sunflower protein that was extracted under similar pH and analyzed under a similar gel protein content. And so there is maybe a place for QPI for a gelling application in the future. Thank you.
Thank you. All right, question.
Did they do flavor? You were talking about the bitterness, but I might have missed that. Did it look like some kind of sensory impact?
No, they didn't do any sensory. So I guess like commonly, quinoa has like two main varieties, and it's just like bitter quinoa is like one, and I guess that was like the more common variety. But I guess also their university was also working on breeding a sweet quinoa that can be grown, I guess, in Europe commercially. So then that's kind of one reason why they were looking at the sweet quinoa. Also, because I guess when they had looked at the study that removed the bitter compounds, it affected the functionality of the QPI. So I guess they wanted to know, like, okay, is the sweet quinoa going to perform worse than just the bitter quinoa, or is it going to perform the same? Does the saponins have an effect? So more so looking at it from that perspective rather than the sensory. Yeah.
Please, again, I don't know if you mentioned or missed that. What is the relationship between the high pH and the decreased purity?
To be quite honest, I don't really know because they didn't really discuss that, and I couldn't really figure out a reason why that would be the trend. I would say, though, that that trend seemed to be unique than the other studies that had happened, because I guess they had also shown that there would be an increase of purity and yield with the extraction pH.
And what is
But
the protein purity and the protein yield?
so purity is like, oh, should I go back?
I had the same question. I think we talked about it a little bit.
Mm-mm.
So when.
Just increase
when you
yield.
finish water,
Uh-huh.
and then you add, I know H, increase the pH. And then you do the isoelectric, and then you reduce the pH to 4.6. You go from 11 to 4.6. What happens? NaOH, HCl.
Like
HCl.
formation of salt, so that's going to decrease purity, and they didn't dialyze it more because of that.
Yeah, so they did that, like, they measure ash content.
No. So yeah, that probably, that makes
They're
sense.
generating more salt and doing dialysis.
Yeah, sorry, I didn't think of that.
Why did the
Huh?
dialysis was not a part of?
So what they did instead is they took, like, once they had the protein fraction, they like solubilized it in water and then kind of like washed the protein to try and remove some of that salt, and then they recentrifuged it, decanted like the more salt filled supernatant to kind of, yeah, but they didn't dialyze it.
So they did several washes. but
Did
the
just one
wash-
wash.
Yeah, so the wash might take some of the salt out. Yeah,
It's not like an equilibrium
yeah.
like dialysis would be,
They
so.
would have to do more washes.
And I guess should theoretically do more washes with an increase of extraction pH.
Is it a typical trend though, where if you increase pH, it will lower your purity?
If
If
you
you
don't
did do-
dialyze,
If you did do dialysis, is that not a normal trend?
no, if you dialyze, no, it shouldn't be a trend.
You want to explain for me the protein yield and the,
Yeah,
what
what's
is
the
what was
difference
the difference?
between protein purity?
Oh, yeah. Oh, yeah. Sorry. So, the protein yield is how much protein you're able to recover from the starting material, and protein purity is how much you're able to, oh, I didn't go far enough. Okay. How much you're able to then concentrate that, right? So then when you have an isolate, you want a purity of around 90% because that's what makes an isolate. So that's just the content of protein on a dry weight basis. Versus yield is like how much you're able to actually extract from the flour itself. So let me go back real quick.
Yeah, but I'll quickly.
Yeah.
Say that, let's say you have 100 grams of flour and 20% protein. So out of that 100 grams of flour, you have 20 grams of protein. But then you were able only to get out 10 grams of protein, so your need is 50%. So you were able to get 10 out of the 20 grams of protein.
So like this is like kind of like this graph here is they measured the percent protein loss. So the dark one is due to the alkalization. So this is how much protein was still left in the fiber and starch, like after they removed the protein. And then the second bar is how much they lost to precipitating. So when they precipitated the protein, centrifuged the pellet, how much protein was left in that supernatant that they like lost during that step. And then this little bar down here is how much they lost during that washing step. So again, rehydrating the protein.
Generally, it should be that the protein purity should be less than the protein yield.
No, so the yield is usually less than the purity.
Yield is
Mm-hmm. Yeah, so then this is the yield, and then that's the purity.
Is that a really low yield, like in general, for this type of product?
I mean, I don't know exactly what the other previous studies had done, but I guess in my personal experience, at least dealing with like legumes and like oat being another, even though this is a pseudo cereal, I guess is more related to cereals, that I consider this a pretty low. Because I feel like yeah, like it is like because this is like well probably like 35%. Like if I'm doing oat on benchtop, like you're getting like 70%. So, and then if their purity is so high too, that means that they didn't have that much ash. Like they had ash, we don't know for sure, but it wasn't like probably like a whole bunch. I mean, don't know for sure because I don't see the percentages, and they also didn't do ash or moisture or fat, so don't really know the composition. Sorry. Yeah, sure.
I guess back to this slide and the one before this, I think. So I think you said previous literature said increasing pH reduced solubility. Or was that,
Increasing pH, reduced solubility. Yeah.
wouldn't you expect the opposite? And like, I guess, isn't this also showing the opposite?
So this is solubility of like the native protein, right? And so by like, I guess undergoing the extraction condition, pH 11 is a fairly harsh, I guess, pH extraction, which would then induce denaturation of like those proteins.
Yeah.
And then again, I feel like also them holding it at this pH for 20 hours probably made it even worse. And so then that's why you saw in the SDS page gel that they probably formed a lot of insoluble aggregates as a result of that, because the protein, and also by the DSC, like that harsh condition denatured the protein basically and almost entirely. So then because it's forming these like insoluble aggregates, and probably also the exposure of more like hydrophobic regions, would the resulting protein isolate won't have as great of interactions with water and would have not as great solubility.
So, but
Let me just clarify what you said is correct. So when you have native protein in the flour and you increase the pH, you get more of the protein
Yeah.
out. Once it's out in solution, it's still exposed with this high pH and then reducing the pH. So the charges, when you reduce the pH, will go away, and then it's denatured protein already. Then you start forming these soluble aggregates.
so when you said it increasing pH reduces solubility, you meant solubility of like the ice.
Yeah.
Ice,
The
not
ice,
the
yeah.
actual.
Yeah.
Mm-hmm.
All right, we need to hold your question for later, but we need to move on. Okay,
Oh.
thank you again.
I'll let you come up.
All right, so Medora, good job, but I want to encourage everybody to try to reduce the time because I also have to do lecture. So compromise, maybe 12 minutes is okay, but definitely not close to 20 minutes. Come on. I know you're perfectionist, Medora, and you want to make sure you say everything.
Yeah, well, because then you were like, okay, make sure to liken it to class too. I was like,
I
well,
know,
make sure was
I
hitting
know.
those points.
It's the art of presenting.
Yeah,
Okay.
and really driving
Can I
it
get
home.
presenter view for Zoom or no?
Oh, you want to use your computer?
No, like, can we do presenter view? Like, there's like notes here and like.
Yeah, mine, what do you?
It is.
Oh,
It is.
perfect. Oh,
There
nice.
it is.
Awesome.
Yeah. All right, and then this is to move.
Cool. Hi, my name is Vici, and today I'll be presenting a paper titled Edible Insect Protein Concentrates, Optimized Assisted Extraction Methods Evaluation. So this paper is from Poland. It's published in the Journal of Food Chemistry, and it's pretty new, published late 2024. Okay, so growing population calls for alternative protein sources for both human and feed consumptions, and insects emerge as a sustainable options. Why? Because it uses a smaller land footprint than compared to animal and plant protein. It has reduced water usage, and it also contains high nutritional values. So it's been reported that 40 to 50% protein dry bases are found in insects. It also has essential minerals and vitamins, good fatty acid profile, good digestibility, making it really promising for alternative protein. And what's the current state? There's a growing interest of insect-enriched products, especially in European Union, where they just recently approved insects for human consumption in 2021. However, there are still challenges in efficient extraction methods that preserves protein quality and functional properties. So this paper hypothesized that salt-assisted extraction techniques can improve protein yield, functionality, and process efficiency. And you see like this graph in class where salting in helps, but a little bit of concentration of salt helps increase solubility, and salting out reduces solubility because of the electrostatic repulsion. The objective of this paper is to compare salt-assisted techniques to traditional, such as alkaline extraction and acid precipitation techniques in several parameters, such as protein quality, yield, characterization, and structure function of insect proteins. So talking about experimental design, there are 3 insects observed in this study: mealworm, house crickets, and migratory locusts, all of which are approved for human consumption in Europe. And then they went through 24-hour fasting period to discard their bowel content before they were freeze-dried and ground by mortar and pestles. They went through defatting steps with a hexane, I think for 3 times, and then they evaporated the hexane under laminar flow hood, constant airflow at room temperature overnight. After that, they further reduce the particle size before vacuum packing it and storing it in negative 20 degrees Celsius. So your defatted insect flour then go through four different extraction methods. I'll go through each one of them. So the first one is control. So no salt involved. It's just alkaline extraction and acid precipitation. The second one is the salting in. So using sodium chloride to increase the solubility of the protein before going through alkaline extraction and acid precipitation. The third method is salting out. So your same control variables, alkaline extraction, acid precipitation, then salting out using ammonium sulfate. The fourth method, the fourth method is a combination of the previous salt-assisted method. So combination of method 2 and 3. So first you have use salt in using sodium chloride, you do alkaline extraction, acid precipitation before salting out with the same ammonium sulfate with the same concentration. Okay. After your salting salt-assisted extraction methods, you do a series of protein characterizations. So they look into the molecular weight distribution using SDS page, secondary structure using FTIR, rheological using rheometer, zeta potential, particle size, amino acids profile using HPLC, and also a series of functional properties tests like solubility, emulsion activity, emulsion stability, water holding, oil holding, foam capacity, and foam stability. Now onto the results. In terms of extraction efficiency, the first method, which is the control method, no salt involved, it resulted in lowest efficiency, as you can see from the lowest by the lowest yield. However, the fourth method, which involved both the salting in and salting out, resulted in highest yield. It's over like 46% of recovery, and this kind of shows related to the class materials where salting in really can improve protein recovery because it increases the protein solubility before going through the next extraction techniques, and also kind of highlights the synergy effects between the 2 salts involved, the sodium chloride for salting in and ammonium sulfates for salting out. In terms of protein purity, so the same table, method 3, which is the salting out methods using ammonium sulfate, results in highest protein purity, over 74%. And this was reported, ammonium ions was reported to be the main driver of this trend because it stabilizes protein structure well by retaining small soluble proteins to prevent protein leaching out of solution. In terms of functionality, again, the method 4, the hybrid method involving both salting in and salting out, resulted in highest functionality by having highest protein solubility over 60% for all the insects observed. Zeta potential. Method 4 shows the highest zeta potentials for all insects observed, and as we learned in class, higher zeta potential relates to greater electrostatic repulsion, which makes the protein less prone to aggregation. And this further confirms the salt synergy mentioned in the previous results table, and it kind of like aligns with the high solubility we see from the salting in and salting out methods. And then next one is secondary structure by FTIR. So from this kind of like proportion of secondary structure found in different all the samples, it shows that extraction conditions do not denature or unfold proteins. So secondary structure are still pretty well preserved. And also there's a pretty good balance in alpha helix and beta sheet ratio in all samples. Although in method 4, it shows higher proportion of exposed functional groups, such as amine, carboxyl, hydroxyl, sulfhydryl, and aromatic rings, which can further play a role in functionality and also like protein ingredient interaction with this functional groups kind of exposed. So to conclude, this paper showed that salt-assisted extraction methods improve protein yield, improve functionality, preserve secondary structures, preserve amino acids content, and also they're like overall a promising technique to increase insect protein potential for food grade applications. And future directions is obviously further identification and characterization of other present components in insect protein. And the big one is chitin, which is contained in the exoskeleton of the insects, which is present in the insect protein concentrate. Also further exploration on different techniques to enhance protein purity. So going from concentrates to isolate. The strength of this paper, obviously, gives you a holistic characterization. So it looks into different extraction techniques and different extraction techniques for different insects, and gives you like a look of their individual structure, function, rheology, and also composition. So it's like a very holistic view of different techniques. And also they employ response surface methodology into extraction conditions, specifically the pH of alkaline extraction and acid precipitation. I didn't show it here because of time, but for different insects, they have like their pH of alkaline extraction and acid precipitations are not randomly chosen. So like they optimize it using RSM based on protein yield, protein purity, and protein solubility. So they went further into extraction optimization, which is like great to see. However, some weaknesses of this paper, I'll break it down into the protein extraction methods. There is no mention of dialysis after any salt treatment and pH adjusted extractions. Obviously, after like changing all the pH, adding salt in, salt out, there's definitely a significant salt content, and obviously this will impact your functionality measurements. The solubility is great, but is it because of the protein or is it because of salt? So they don't mention dialysis; they just mention to wash with DI water, which is obviously not enough, and they also didn't include any ash content. So kind of like the same issue, the paper that Medora presented. And then for me personally, the salting out method was unclear, particularly method 3 and 4. I wasn't clear if they add the ammonium sulfates to the supernatant of acid precipitation to further recover the protein, or they did the salting out and acid precipitation methods the same time. So this would kind of like interfere with a replicability of this study, and can other paper like replicate the results, but they didn't like fully clarify. And also they didn't mention of adjusting pH of the final concentrates before doing all the protein characterization. So yeah, in terms of like food applicability, because a lot of the food application happens in neutral pH, but they didn't really mention of adjusting it back to pH, which kind of like, like how translatable is the data for like food application. And then in terms of the protein characterization methods, they do acid hydrolysis, only acid hydrolysis for amino acid composition. So that's why I didn't show it here because of time, but it only showed that 17 amino acids were identified. And obviously a lot of them are underestimated because they do only acid hydrolysis. Zeta potential looked really great, but it was measured at pH 8.4 instead of neutral. And they didn't really explain as to why, which is not the case for like food application. A lot of the things happen in neutral pH. So they didn't really explain why they measured it at pH 8.4. And then it's they very briefly mention how structure like amino acid compositions, molecular weight, secondary structure, rheology relates to functionality. They just say that it impacts functionality, but they didn't really like explain the mechanism of how. And also they didn't really call out specific functionality. They didn't really call out specific food application, which is kind of a bummer. But yeah, they don't really mention it. And also a fatal one. It's a little cut. But they mentioned foam capacity and stability in graphical abstract, but there's no data on that. And I'll show you the results. In the results table, they have like the abbreviation for foam capacity and foam stability, but no data. I just found out this morning. Yeah. So yeah.
That's something you could probably like contact them, right? Like somebody you have the like you should probably contact them or no?
Yeah.
Contact is published already.
Yeah. So yeah, pretty fatal and like interesting, but yeah.
So bad.
But yeah, thank you. Overall a good paper for like learning insect protein concentrate. I think it's so interesting.
The data there is in other parts. For example, they in their website. You
Not
can access online to that. Maybe some of the papers, the extra data publishing.
not even in like not in the discussion, because like yeah, like some discussion like mentioned supplemental. Yeah, it's really gone. And like I was looking at the abstract and like let me go back a little bit. Oh yeah.
But tell you what could have happened. Submitted the paper and then the reviewer said remove foaming.
Yeah. I was
But
like,
they already
yeah.
have the graphical abstract, they did it edit,
Mm-hmm.
and they forgot to edit the footnotes in the
Oh,
paper.
okay.
And I'll tell you what, they didn't have Pam as an advisor. Because
Yeah.
I catch these things.
I was like, what is FC and FS? Like when I was looking at the graphical abstract, and I did a little
Yeah.
digging and like, bummer.
Chemistry takes forever to publish it.
Yeah,
Longer time, so by that time student might have gone from lab.
I know, yeah, there
Oh.
you go. Uh-huh.
Oh no.
Yeah, I have two questions, from two questions. real quick.
Well, first of all, let's thank her.
Yeah, let me go back.
Okay, go ahead.
Okay, yeah, it was a really interesting topic. I know that, well, for the slides that I downloaded, you had like a slide who was mentioning about the amino acids.
Yes.
And, I was just, curious because that's actually a question that I had, it was mentioning there that you have, 40 to 46% of, hydrophobic amino acid.
Yeah, it's mhm.
And I was just wondering, what does that mean? Like, is it like a high percentage?
It's not, I don't think they discuss like if it's like high or like expected. They just like kind of like sum it and like just reported the number. But they did mention that hydrophobic amino acids like does relate to like increased oil absorption, like kind of like we learned in class. But yeah, they didn't really kind of like go into further, like go further into like what does it mean? Is it like higher or like lower?
Yeah,
But it's like pretty
you
yeah.
guys think that.
Yeah.
Also, they only did amino acid with 17 amino acids, so
Yeah.
that 40% is from the 17.
Yeah.
Yeah.
Probably this, it's not probably
Yeah.
true.
Oh, okay, yeah, because they only did the acid hydrolysis. They didn't do base hydrolysis and digestion. So a lot of the numbers might be underreported. Yeah.
Or
One last question. So based on the functionality, the cell solubility 60%, something like that. What would be an application that you could think about?
also, I think like high salt content, I would assume that this is like very high salt because all the pH adjustment, like salt extraction methods, like, well, is it like is the high solubility good for by the protein or by the salt? But I mean, I think like what is out there in terms of like insect protein is not beverages, like it's not like insect beverages, but a lot of it's just kind of like pasta, which I think like it would help with like I don't know if it would help the dough, but yeah, I wish like they also meant like specifically call out what food application this might be in because like they seem to have more than enough data. They also measure like viscosity, rheology, which I didn't include because of time, but they seem to have a pretty good data to like can make a decision of like what this insect protein is good for, but they didn't in their paper.
How
So
much
that's
is
all there.
solubility percentage? What concentration?
That's a good question.
Yeah.
I know it's not in DI water as well, so that's interesting. They did it in like phosphate. I think like I put it here in like
It's not uncommon, put it at the
Okay.
buffer, but what's the protein?
I can look.
0.1%.
Oh, thanks. Oh, is it really?
I think it's 0.1%.
Thank you.
Was dispersed and whatever of 0.1.
Oh yeah, you're right. You're right. Yes.
What is the
It's
buffer?
0.1%, yes.
It's
like
so low. That's your 1 milligram per ml kind of thing.
That's
Yeah.
not solid.
Yeah.
Oh.
Thank you. Yeah.
Does it make sense, you guys?
No.
No, it doesn't.
Even for Medora's paper also was 1%.
It was 1%, so I was like, at least that's something, like not point something, but it was still a little low.
It's 10 times more than the point.
Yeah.
Yeah. Other questions? Okay, well, thank you.
Thank you.
All right. So first of all, I want to say very good presentations and coverage and figuring out the weaknesses and linking to what we studied in class. I just want to say you were polar opposite in timing and speed. I think we have to find a compromise. Maybe 10 minutes is too short. You had to rush through your presentation. And I'm evaluating one thing and you were 3 steps ahead of me. So, okay, 5. I don't know what she said. Yeah, and Medora detail oriented. So should we say, what do we want to do? Huh? 12 minutes? 15?
I don't, how long was mine?
It's you're close to 20.
So wait.
Yeah, I was looking at the time.
Cause when I practiced, I was at like 15, and
Yeah.
I said less days today, so.
Right.
Yeah.
I thought it was definitely over 15 because I looked at the time. Anyway, so 12 minutes, should we? 15? You agree, Sarah? Yeah, but you're not doing a presentation. Okay, 50. So we won't ding you too much for if you took out a lot of points for Medora, give her back a point. And yeah. So it's good to set the stage. Very good layout of your presentation. So good job. Okay. All right. Go back to enzymes. I have to finish enzymes today. So we're having dinner here today. We ordered pizza. What? I have 15 minutes. Go. Okay, thank you, sir. Ah. All right. So let's do a quick recap. Our food enzymes, proteins, you guys.
No. Yes.
No.
Yes.
Food proteins are, but not all of them. Okay, got you. We are still April Fools, so it's fine. Okay, yes. Quick definition of enzyme, very quick.
in their definition, you can state their function? Come on, you can do it. You can do it, Shri. You're presenting next week.
It's biological catalyst that
Thank you. Teamwork.
speeds up a reaction without like being so.
Very good, very good. Biological catalyst, by definition, it reduces the activation energy and helps convert reactants to product. Do you remember any of the classes of the enzyme? Just mention one or two. The most common that you know of. Good job, but what it belongs under a class? Proteases belong under a class. So lactase, protease, lipase, hydrolase. What else? Do you remember another one that's very common other than ligase? Okay, it's not very common, but yes, ligase. Another one, what's polyphenol oxidoreductase? So we're going to talk a lot about hydrolases and oxidoreductases. There's also transferases, isomerases. So these are important enzymes classes. Okay. Very quickly, what's the induced fit theory? So we talked about lock and key theory. What's the induced fit? Do you remember? Is it really a rigid spot where the enzyme binds the substrate? So the binding site and the active site. So you'll have the binding site on the substrate, and then you have the active site on the enzyme. Are they rigid? They don't, they're not rigid. So basically the environment impact the binding site, and when the substrate comes to bind with the enzyme, they kind of fit each other to kind of lock each other. So it's not like a solid state. pH, ionic strength, water activity, temperature impact that binding site. So that's to remind you that it takes the shape needed to cause the binding to be effective. Okay. Impacted by environmental conditions. We talked about several applications. I'm not gonna go over them just because of time. I want to move forward, but just always remember examples, because in the test I might ask you, okay, give me an example when you use enzymes to determine quality or to impact quality, or when do you measure enzyme activity, or how do you use enzymes for analysis of constituents? How do you use them as processing aids? So think of these questions when you are studying. Important parameters. Do you remember what Km is? Spent some time last week on Km. Huh? Huh? Oh, because Minton. Okay, but what is? Oh.
The upstream concentration at which the speed is half of the maximum.
Yes, at which the rate is half Vmax. Okay, it's the substrate concentration at Vmax over 2. Okay, and Vmax is when you have the enzyme is saturated with the substrate, you reach Vmax. Importance of enzyme kinetics whenever you want to set up an enzymatic reaction, whether you want it as during processing, or you want to understand how you can inactivate the enzyme, or if you want to set up an analysis where you use enzyme to determine glucose content, for example, you really need to understand kinetics. You need to get the Vmax and the Km characteristics. So we solved the Michaelis-Menten equation last time, and then we converted it to a linear equation so that we can determine Vmax and Km. And the linear equation is called Lineweaver-Burke equation. Names of people, basically. And it's just a reminder that Km is a determinant of efficiency, affinity. So the higher the Km, the lower the affinity, because you would need higher substrate concentration to reach the same velocity. So lower Km, higher affinity of the enzyme to that particular substrate. We define Km, types of inhibitors. So also you want to remember the differences. We have irreversible inhibitors and reversible inhibitors. Under reversible inhibitor, we talked about competitive inhibitors, non-competitive, and you like that very much, uncompetitive inhibition. So also remember the difference between those three. What does this figure tell us? So here we have the generation of product, here we have time, and then here we have enzyme concentration. So lowest enzyme concentration and highest enzyme concentration. So whenever we are trying to use an enzymatic reaction, we want to select the time where we can control and be able to measure whatever we're measuring, absorbance or what have you. But also we need to pick enzyme concentration where the reaction is linear. So you have an increase in product over time, the reaction is linear. So you want to select the time and enzyme combination that gives you good prediction. If you select enzyme concentration 4, let's say A is 10 minutes, definitely you're not in the good prediction area. So some factors that affect the rate of enzymatic reaction: concentration of the enzyme, concentration and type of substrate, presence of inhibitors. The list is long. So these are questions that will help you after you study, see if you can answer them. Okay, so I'm going to move ahead. If you have questions, you can ask me. If you want pizza, you can ask questions and we can sit down and talk more. No questions. All right. So this is actually an application to understand native enzyme systems in milk. I did that also during my postdoc. I was looking at the native enzyme in milk, which is plasmin. It hydrolyzes beta-casein, and it does lead to issues with milk thinning and gelation over time. So, but also milk has natural inhibitors of plasmin, of that enzyme. So we were trying to characterize these inhibitors to understand how we can inhibit a breakdown of beta-casein in milk. So we did the reaction. We collected the initial velocity at different substrate concentrations, and we plotted Lineweaver-Burk plot. And we got, if you remember, the intercept is the same, but if you bring down those lines, you will have different Km. What does that mean? Do you remember if this is competitive, non-competitive, or uncompetitive inhibition? You can look back. So what is it? It's a competitive inhibition. So in this case, the enzyme, you have a substrate, say beta-casein, and you have an inhibitor that is binding in the same binding site with the enzyme. So the rate of the reaction is not impacted because once the enzyme binds to the substrate, the reaction proceeds at the same rate. So what's happening is the inhibitor is competing with the substrate. So then when we did the reaction and we plotted it, and then did some calculations of Km and Vmax, catalytic efficiency, which is Vmax over total enzyme, and then you have Kcat over Km. So the lower the Kcat over Km, the catalytic efficiency, obviously, the lower is, the more is the inhibition. But what you want to look at is Km changed, but Vmax stayed the same. So with the inhibitor, the Km increased. So here times 10 to the -2, here's times 10 to the -1. So Km increased, Vmax stayed almost same. And then the catalytic efficiency, because the Km increased, it got reduced. So we concluded that the type of inhibition is competitive inhibition. So now how does this apply to a different setup where in this example here, inhibition of, so what are we inhibiting here? We are in polyphenol oxidase. So in this example, we are looking at, not us, somebody else, was looking at no inhibitor with PHBA inhibitor, the green, and with another type of inhibitor. So if you see here, we have two types of inhibition, the competitive inhibitor, the green, and the purple is the non-competitive inhibition. The non-competitive inhibition is when the enzyme and the substrate bind, but then the inhibitor binds to that mix, to that complex. The enzyme substrate complex, the inhibitor binds to that. So it really reduces the rate of the reaction because it's not impacting affinity. The enzyme still binds to the substrate, but once it's bound, it depends on how much inhibitor you have in the system, it can bind and prevent the progression of the reaction, so that impacts the velocity. Okay, so if we look at which inhibitor to use in this case, the preference is for non-competitive inhibition. Because what happens here, no matter what's the substrate concentration, you don't need a lot of inhibitor to reduce the reaction rate, irrespective of the substrate concentration. So when you need less inhibitor, that is better financially. But then there are considerations that you have to take. The cost of the inhibitor, whether or not it's GRAS, generally recognized as safe. Is it legal to use it and no side effects? And does it contribute or impact the sensory perception? So there is always a compromise in choosing your inhibitors if you want to inhibit an enzyme. Okay, so most common enzymatic reactions that we use for analytical purposes, for measurement, are coupled reactions. Some of you know that when you measure total glucose in our lab, it's a coupled reaction. You have one enzyme, you have two enzymes. The first, you convert one substrate into a product, and then you use the product and another enzyme to convert it into another product that you can measure or that absorbs light. So oftentimes you need those two reactions to get to a product where you can measure absorbance. So we call this first reaction in a coupled reaction, we call it the measuring reaction or auxiliary reaction. The second one is the indicating or indicator reaction, because that's what you measure absorption and the indicator. But before I go there, I forgot, what's the definition of 1 unit of activity versus concentration of the enzyme? When I say concentration of the enzyme, what am I really measuring? Just concentration. What am I measuring? What are enzymes? proteins. So I'm measuring the percentage of or concentration in milligrams per milliliter or whatever it is, nanograms in milliliter, that's concentration based on amount of protein. But what is the unit of that? It's a good college board question. Yeah? Remember? You know? Huh? Well, it is, well, yeah, there is time involved. Yeah.
Concentration. Products run over some time.
Usually it's product or substrate, but usually more commonly it's the amount of substrate converted, or probably millimole, micromole even. Micromole of substrate that is converted per minute under specific conditions of the reaction. So, and usually the unit of the enzyme is dependent on the substrate. So sometimes when you buy an enzyme and they give you the unit in that spec sheet, they have to tell you what's the substrate that they use to measure that unit, and they have to say what the pH and temperature was. So that you know, okay, at pH 7, at 37 degrees Celsius, I'm getting 5 micromole per minute, so that would be 5 minutes. Okay, I'm converting 5 micromole, converting 5 micromole of substrate per minute.
It's not on the,
Huh?
sorry, it's not on the product, how much they.
It's some use the products, if it's easier to measure the product and they can convert it to product, they can, but this is the official, not the substrate.
I think this is a college board question. This is a college board question.
It is, yeah. Yeah, that's what I said, college board question.
Because I remember you had said, like, actually, it also depends on the.
Yeah. Well,
Substrate.
oh, there is sob story. There is about that college board question. I always thought it as at specific conditions. We were at nationals and the question came and our student answered correctly. And they marked it wrong because on the database, it didn't say under specific conditions. So they were wrong, but I couldn't challenge for them. I'm sitting in the audience. I couldn't jump and say, no, wrong. The answer is wrong. Anyway, but this is what it is. If you get a specification sheet and you would, for an enzyme, you will see under the units, they specify the conditions. And they specify the substrate because every substrate will elicit different activity units. Okay. Oh, shit. If I'm measuring enzyme activity, should the substrate be much greater than Km or much lower than Km? If I'm measuring enzyme activity. Think about it. Do you want the reaction to be dependent on the substrate or to be dependent on the enzyme concentration?
Much greater.
Huh?
It's going to be greater, so that's dependent on that.
Yes, so you want it to be first order with respect to the enzyme. So when you change the enzyme concentration, you want the assay to be sensitive to that. So you saturate the insistent with your substrate. So it will be 0 order with respect to substrate. So your substrate concentration should be much greater than Km. All right, there's an example of coupled reaction. So here you have, you're measuring glucose. This is a little different than what we use in the lab, the 2 enzymes. Here we have hexokinase for the first reaction, which is a transferase. You take 1 phosphate group and give it to the glucose. So you get glucose 6-phosphate and ADP. So, and then here you take the glucose 6-phosphate, which is the product here, and react it with NADP And the second enzyme is glucose 6-phosphate dehydrogenase. So you're removing a hydrogen. And giving the NADP, you're giving it the hydrogen. And it's also a transferase. So this product right here is what absorbs light and measurable. So you would measure this product. Okay. So we said we have 6 classes, but I'll be talking about 2 important classes in foods. There, we can't talk about all of them. Transferase and isomerase are important, but the biggest classes are hydrolases and oxidoreductase. So we're going to talk about some examples of all of these guys in food application. And also we're going to talk about polyphenol oxidase quite a bit. And just a couple of examples where you use these 2 enzymes. What's the definition of hydrolases? What do they do and how do they work? Hydro. Say that again. Not necessarily peptide, protease, yes, but hydrolases will hydrolyze. There are different types of hydrolases. Protease is one, lipase is another, lactase is another, sucrase or invertase is another. So hydro, what's the role of water? So when you break the bond, the parts of water, OH and H, become part of the products. They get absorbed by the products. So the elements of water go to the broken bonds. So triacylglycerol or TG, triglyceride with lipase and water will give you 1 fatty acid and diacylglycerol. Break another fatty acid, monoacylglycerol, and 2 fatty acid, or glycerol and 3 fatty acid. And each time, each fatty acid you break off from the triglyceride, 1 water molecule is taken up. So if you break it completely into glycerol and 3 fatty acid, you need 3 molecules of water. So pectinases are important for different applications. So you have different classes of pectinases. So if you remember, pectin is have glucuronic groups, glucuronic acid groups. Some of these are have a methoxy group attached to it, and some don't have a COO minus. So the number of methoxy groups attached determine how well it can form a gel. So if you have less than 50% esterification with methoxy groups, that means you can form a gel because you have COO minus, and if you have divalent ions, it helps with the formation of the gels. So pectin will polymerize with having divalent ion in there. So the higher the degree of esterification, the lower is the gelling ability. So sometimes you add an enzyme to break off the methoxy group. So when you break off the methoxy group, then you can enhance the gelation properties of pectin. So the enzyme that breaks off methoxy group is pectin methyl esterases. So it breaks off the esterified methoxy group. Okay, so you have also pectinases that break the glycosidic bond. So you have polymethyl galacturonase and polygalacturonase. So 2 different ones. One will cut after a group that is has a methoxy, and one will cut after a group that doesn't have a methoxy group. So that's why you have two enzymes here. I don't know why this is cutting. We did that one time, right? I don't know why. So it's used in the food industry to clarify apple juice, for example. Because when you use these pectinases, you break down the pectin and then you clarify your apple juice. That's one application. The pectin methyl esterase, for example, in juice, it's naturally present. So you have to pasteurize to inactivate it. If you don't inactivate it and you have pulp, then you see that separation over time in your orange juice, because what happens is those native pectin methyl esterases are going to break those methoxy groups off. And if you have calcium or divalent ions, they're going to form polymers and sediment. And then you have orange juice separation of your pulp. So there has been studies to inactivate pectin methyl esterases to using means other than just heating to preserve the quality of the juice. One was looking at using high pressure combined with thermal treatments, but at low temperatures. And then here, they use the inactivation kinetics. In this case, they measure the reaction rate constant, and they measure the D value. In this case, it is the time needed to have 90% reduction in the activity on the reaction, 90% reduction in the reaction. Okay, so here they looked at the different temperatures and the different pressure. And they found, I think I animated, no, I didn't animate. So they found that this highest one with the highest temperature with the highest pressure increased the rate constant. And then the time to get 90% inactivation was reduced significantly from 116, 17 to 4.6 minutes. So in 4.6 minutes, they were able to reach 90% inactivation. Okay. Amylases are commonly used to produce from starch. What do you produce? Huh? Maltodextrin, yes. And so high fructose corn syrup. Because for producing high fructose corn syrup, you want to break everything into glucose. So you would use, if you gelatinize the starch, you would use alpha amylase and glucoamylase. Alpha amylase will be an endo enzyme that will break inside and can also break alpha-1,6 linkages, alpha-1,4 and alpha-1,6, and glucoamylase will break one at a time. And then when you have that glucose formed, then you can use isomerase to produce fructose, high fructose corn syrup. But all of this, the alpha amylase and glucoamylase are hydrolases. This is another class. Isomerase is the class. This, to get glucose to fructose, you need an isomerase. Okay. Lactase is another one. And in lactase, it's used for most commonly milk free of lactose. So for those people that are lactose intolerant. So what is another name for lactase called for people? Huh? Galactase. Galactase. Galactose. Huh?
It's just galactase, right?
Yeah, I thought so. Yeah. You can say galactose, it's fine. Yeah. Yeah, so, yeah, so that's a common application for this hydraulic, type of hydrolysate. Hydrolysate, what? Can't say this anymore. Hydrolases. All right, it's late. And then sucrase is also known as invertase because it inverts sucrose into glucose and fructose. Are you familiar with this candy? What happens? Yeah, I dislike them too. So, you know, what happens here is they actually add enzyme during the production, invertase, so that they're using sucrose, let's say, and then if they don't put the enzyme, what happens over time with the sucrose? Crystallizes. So the invertase slowly convert the sucrose into glucose and fructose, which don't crystallize during storage. And then that's why you have that syrup thing inside. So.
Why they don't have acid instead of.
Acid?
Yeah.
Do you want to eat it if it's that
No,
sour?
not too much, but just a bit, for example, lemon or others.
Maybe
Can
they didn't
control
want to
the
make it
taste.
sour. The cherry is already sour. I have no idea. To be honest, like Medora said, I really dislike them too.
Be better if it was.
Yeah, well, okay. But this is just the application of the invertase is to prevent the sucrose from becoming crystalline over time. All right, and then we have your favorite protease is also a hydrolase and is commonly used to sometimes gluten, they add protease to reduce the elasticity, enhance the viscosity. So sometimes it's used to for meat tenderization, sometimes obviously cheese formation. Aging of cheese to give you that characteristic flavor. Many applications for proteases, and a big part of production of protein hydrolysates ingredient is using proteases in the market. Okay. There are different proteases that can be used with different activity. Some are specific, very specific, like trypsin, only breaks after lysine and arginine. Some are very common, non-specific enzyme, like pepsin, for example. Lipase is another one used in the industry to break down triglycerides into monoacylglycerol and diacylglycerol for production of emulsifiers, basically, and used in bakery products and many other to enhance emulsion. Okay, so oxidoreductases. So here, what is the definition of oxidoreductases? Tired? There we go. Huh? Yeah, well, it's transfer of electrons and hydrogens in the reaction. So catalase or peroxidase breaks down peroxide into water and oxygen. So if you have peroxides that you want to get rid of, you add catalase or peroxidase to get rid of peroxides in your food or ingredient. The most common one that is very problematic is polyphenol oxidase because close to 60% of the produce are lost because of browning. So it is undesirable color, unappealing. That's why it's problematic, and it is commonly endogenous to a lot of different types of produce or foods. But sometimes it is desirable, like in tea, the browning of tea is based on polyphenol oxidase enzymatic activity, but also shrimp can become this color. I mean, of course, if you see it like that in the market, will you buy it? No, it's not necessarily bad for you, but it doesn't look appealing. And of course, the produce, the fruits, and then the Granny Smith, this was also a College Bowl question. Why does Granny Smith, when you cut it, it doesn't brown as fast as any other apple? Huh? Genetically modified. My understanding was genetically modified to have less polyphenol oxidase. Somebody told me, or either in the answer of the College Bowl, that the substrate genetically modified to have less phenolics. And I still feel like it's genetically modified to have less polyphenol oxidase, but look it up at +2. Come back next week and tell me. All right, so the reaction is you have your phenolics, and then you have different types of polyphenol oxidases, not just one enzyme. One will convert it to diphenol, and then another will convert it to quinone. This is the precursor of melanin. So this gets polymerized, complex brown polymers formation. These are your melanin. If you reach to quinone, you can reduce it back. But once you reach melanin, it's not reversible. So you can reduce it back if you add ascorbic acid or a reducing agent like thiol-containing compounds or sodium sulfite, for example. Sodium sulfite is a reducing agent that you can use for food application. Cysteine is a reducing agent that you can use in food application, or acid. Sometimes if you put a little bit of lemon on your apple, you prevent it from getting brown in color. Thiol containing, it has SH. So like cysteine has an SH group, sulfide group. It acts as a reducing agent. Yeah. And then you can also add like sulfur dioxide, you can cause an adduct formation. I'm sorry, you can see it, but you form a product that prevent the formation of the polymerization. But you have it in your notes. But there are other ways that we control polyphenol oxidase. Of course, heating to inactivate the enzyme, dehydration. Of course, you know, enzymatic reaction require water. If you dry your fruits, you prevent the browning. Reducing the pH, adding a reducing agent, chelating agent, because oxidation reaction requires metal. So if you add chelating agent, you slow down the reaction. And then you add aromatic inhibitors so that the enzyme will react with the inhibitor instead of reacting with the actual substrate. Or you can modify the atmosphere in your package, removing air, putting argon, for example. Genetic engineering. There we go. It is the
Yeah.
You found? College Bowl questions. I'm right.
For 90%.
Yeah, 90%. Plus 2. Okay. Promising. Promises. I make promises. I make sure we justify. Okay. Irradiation, also high pressure processing. So a lot of different ways to inactivate polyphenol oxidase. So one example, and it's also a paper that I have on Canvas for you to look at. So here what they looked at to determine two varieties of peaches. And they wanted to understand the enzyme kinetics to see how they can inhibit or use these varieties in different applications. So they got extracted the enzyme from each of each variety and determined their specificity to different substrate and also determined inhibitors for these. So they did the kinetics, calculated your Vmax, Km, and also the catalytic efficiency, and plotted Lineweaver-Burk plot. And well, plotted the plot and then calculated, and then they determined that for one enzyme, they differed in the substrate. So one enzyme had this yellow box, the other enzyme wanted this other substrate. And then they looked at inhibition, and they determined these are all competitive inhibitors because of similar intercept, different Km. You can also look at the table with the values, and they determined that this particular benzaldehyde is the most effective inhibitor. I think this is my last example. The other case study here, they looked at, active packaging, so changing the environment in the package to replace air by argon, and determined how that works. Apparently, the presence of argon is a competitor to oxygen, and you can see here your Vmax does not change for these two types of polyphenol oxidase, but the Km value in the presence of argon increases in both cases. Oh, I lied, not my final thing. Of course, food analysis is my second favorite topic. And of course, enzymes and food analysis, I'm in heaven, right? So food analysis, enzymes, proteins. So we use enzymes in analysis. And an application here with the use of enzyme is another type of two reactions, two enzymes, glucose oxidase and peroxidase. So these are the enzymes that we actually use in our lab to determine glucose. So the first one converts the glucose into gluconolactone, but that is not our product that we care about. The peroxide here that reacts with a dye, it oxidizes the dye, and the oxidized dye has color. So using glucose oxidase also is used as an oxygen scavenger in packaging. So it's covalently attached to an insoluble support, or it's absorbed via hydrophobic interaction. But when it's there, it kind of scavenges the oxygen. And this is how you extend the shelf life of your products that are prone to oxidation. Okay. So where do we use immobilized enzymes? We have an idea. Very common use. What? ELISA, yes, absolutely, that's one, of course. So your enzyme, ELISA stands for enzyme linked immunosorbent acid. Enzyme. Yeah, ELISA is one application. What else? Very common. Oh, yes, very good. You can have your enzyme there if you are trying to purify inhibitors, for example. Yeah, okay. What else? Food application. Very common. What do we do with lactose in milk? Do they immobilize? But they pass the milk through membranes where you have the enzyme immobilized to break down lactose by lactase. So Oh, high fructose corn syrup and production of hydrolysis. So this way in industry, they can regenerate the enzyme and use it. Instead of batch processing, you lose the enzyme. If they immobilize it, they can utilize it multiple times. So enzymes in baking, sometimes you use a little bit of amylase so that the starter or the fermentation process, your microorganism will have more sugar to help with the fermentation process. Break down gluten a little bit to enhance viscosity. Glutamyl transferase is your transglutaminase. So it's just sometimes people refer to it as glutamyl transferase, but it's really your transglutaminase. And it helps with polymerization and enhancing, for example, the formation of rice bread. Sulfhydryl oxidase, it induces disulfide interchange, strengthen weak doughs, enhances elasticity. That's your glutamyl transferase, transglutaminase in rice bread. So it deamidates here and causes a covalent linkages. So transglutaminase, this is your glutamine, this is the carboxyamide group, an amine is lost, and the carboxyamide group or the carbonyl here reacts with the amine. You have a covalent bond, and that is intermolecular. Enhancing solubility, we use not transglutaminase, we use glutaminase in our lab to help extract proteins from matrices where the protein is not soluble, like hemp, for example. Hemp protein is not soluble. Glutaminase, in this case, doesn't form polymers, not the transglutaminase. It just removes, it's actually a hydrolase. So it just removes, break down this bond and remove an ammonia. And the water is part of, water elements goes to both products. And then instead of having a carboxyamide, you have a carboxyl group that is ionizable. At pH 7, it will be negatively charged. Then you enhance the solubility of the protein. In milk, chymosin for cheese making, proteases for flavor, lipases for flavor, sulfhydryl oxidase to remove cooked flavor, galactosidases is the lactase, that's a lactase, and microbial protease. For some reason, this table has milk, soy milk, and it says here milk and dairy product. I don't know why. See? But removal of unwanted constituents, that's basically specifically targeting constituents that you don't want, like lactose, peroxide, or removing bitter peptides. Okay. Oh, it feels like I'm surprised that there's still more slides. Okay. Here is showing you that we can measure certain constituents using enzymes. Sulfite is important because of sulfite sensitivity. So in order to put on the label whether it's sulfite free or less than a certain parts per million, we can measure sulfite in a product using enzymes, glucose, obviously starch, you break it down all to glucose and then measure glucose. And D-malic acid in nature, this is the acid that is present in apple. It's L-malic acid in apple, the naturally occurring acid. In order to determine if there is any, determine the authenticity of your apple juice, you can measure D-malic acid using enzymatic reaction. And of course, when we're measuring substrate, you don't want the substrate to be saturating the enzyme. So you want the reaction to be first order with respect to the substrate. So you want that linearity with regard to the substrate concentration. So So the substrate much lower than the Km, whereas when you're measuring enzyme activity, your substrate should saturate the enzyme and the reaction should be first order with respect to the enzyme. Peroxidase, lipoxygenase, usually we test them to determine efficiency of blending. Lipoxygenase, we test it to see how much lipid oxidation potentially can happen in our sample. Alpha amylase to determine in flour, the higher activity of alpha amylase, that means the starch will not form viscous solution because alpha amylase would chop on the starch. And then rennet for the formation of cheese. And these are my examples, and I can send you home. No pizza. All right, we can start dairy proteins next week.